My name is Marie Neidem, and I'm a professor at Soka University of America. Today I'm going to be talking to you about the taxonomy of the order Phlebobranchia in class Ascidiaceae. Class Ascidiaceae refers to the ascidians, commonly known as sea squirts, which are invertebrate chordates. In the previous video, I explained how to tell the difference between the three orders of ascidians: Phlebobranchia, Stylidobranchia, and Occlusibranchia. In this particular video, I will walk you through how to tell families and genera of the order Phlebobranchia. And I'd like to acknowledge information and image credit to Gretchen Lambert, Francoise and Claude Monio, and Rosanna Rocha. The first thing to figure out if you're trying to understand the Phlebobranchia is whether your animal is solitary or colonial. And as a review from the previous video, a solitary individual lives alone, lives not attached to any other ascidian, whereas a colonial individual is genetically identical to other zooids living in very close proximity to it. In the Phlebobranchia, there are, in particular, I've just highlighted 5 families. In the top right, you'll see where I am in terms of whether I'm talking about families or genera, etc. But we'll start out with families in the order Phlebobranchia. And there are many more than 5 families, but in the interest of time, I'm talking about what I think are the 5 most common families. Of these 5 families, 4 are solitary, have solitary representatives only, and 1 has colonial representatives only. So if you've determined that your ascidian is a Phlebobranch, and you've determined that it is colonial, there is only 1 family that you have available to you to continue to research, and that's the Paraphoridae. If it's solitary, there are 4 common families: Agnesidae, Ascididae, Sionidae, and Corellidae. Let's first talk about the family Paraphoridae, the colonial Phlebobranchs. And the Paraphoridae have zooids connected by stolons. This is a photograph of Paraphora japonica, and the zooids are the circular yellow structures that you see in this photograph. And then they are connected, you may be able to see, by this network of narrow branching stolons, which are the yellow lines that connect the individuals. In Paraphoridae, the zooids are connected by the stolons. There are 2 genera of Paraphoridae, so now we are distinguishing genera inside the family Paraphoridae, inside the order Phlebobranchia. And they are relatively easy to tell apart, these 2 genera. Actinidea has more than 5 rows of stigmata, and Paraphora has 5 or fewer rows of stigmata. On the left photograph is a picture from Rosanna Rocha of an Actinidea, and the stigmatal rows are in the branchial sac. The branchial sac is the structure in the body of the organism that helps the animal feed and also to breathe. So what we're looking at with these stigmatal rows is how many rows in that mesh-like structure in that branchial sac. And you can, in this really nice photograph here, you can count 1, 2, 3, 4, 5, 6, 7. I see at least 7 rows of stigmata. Again, just the rows, the horizontal rows in that grid-like mesh structure. More than 5 rows of stigmata, Actinidea. In the diagram from Richard Fox on the right-hand side, you see a Paraphora, genus Paraphora, and there's only 4 rows of stigmata drawn here. The 4th row, the posterior-most row, is almost entirely obscured by the gonads in the intestine, but you can see it peeking out just in the second loop of the intestine there. You can see a couple stigmata. So again, this Paraphora 4 rows to stigmata, that's less than or equal to 5. If you have decided you have a Phlebobranchia, a species in the order Phlebobranchia, and it is a solitary individual, meaning living by itself, not attached to other members of its species, and not genetically identical to other members of its species that it's attached to, it's a solitary. And there are 4 solitary families that I've chosen to represent here in this video: Agnesidae, Ascididae, Sionidae, and Corellidae. I'd like to talk now about how to distinguish between those 4 families that can have solitary species. And the goal here is to look at the position of the gut or intestine relative to the branchial sac. So gut and intestine would be referring to the same structure here. If the gut is posterior to the branchial sac, posterior meaning further down on the animal, right, away from the oral and atrial siphons. If the gut is posterior to the branchial sac, the family will be Sionidae. This picture here, this diagram, represents an example of a Siona in that family, Sionidae. And we're looking for the branchial sac, and you can see that mesh-like structure. It takes up the majority of the body, starting just posterior to the oral siphon. Just below or posterior to that branchial sac, so look down after the branchial sac finishes, continue looking down the animal, and you'll see the gut. You'll see the stomach, the intestine, and then moving up to the rectum and the anus. So again, gut posterior to the branchial sac in this Sionidae. Or the gut could be to the left of the branchial sac. So just as a reminder, we want to think about left and right sides of an ascidian relative to the dorsal and ventral sides. So when we think about the endostyle, let's look at this picture C here from Bonnet and Rocha 2011. In picture C on the left side of that photograph, you see the endostyle, which is the very thin pinkish structure that starts right next to the oral siphon and moves down posterior to the end of the animal. That's the endostyle. It creates the mucus net that helps the animal capture its food. If you locate the endostyle, that is the ventral side of the animal, also known in invertebrate biology as the stomach side. The opposite side, where you can see the atrial siphon, so that's the projection, the protuberance, where the animal is going to release sperm and eggs and waste, feces. That is the dorsal side, where you see that atrial siphon there. You might also be able to see that the intestine ends right about at that atrial siphon, so the feces are going to be released to the atrial siphon. The atrial siphon is the ventral side, is the dorsal side. So we think of that in invertebrate biology as the back of the animal. So if you think of the dorsal side as the back, the ventral side as the stomach, then we are looking at, on C, photograph C, we are looking at the left side of the animal. And we are looking straight on at the gut. So the first thing that we see right on the other side of that body wall is the gut. And then the branchial sac is behind it in our view. So in this particular photograph, the gut is to the left of the branchial sac in this animal. In photograph B, the gut is also still to the left of the branchial sac because it's the same individual. But we are now photographing the right side of the animal, so we have to look through the transparent body wall, through the branchial sac to the other side of the animal where we see that gut. So when you photograph the right side, what you're looking at first is the branchial sac, then behind that is going to be the gut. If you see the gut to the left of the branchial sac, like in this particular animal that's photographed by Bonnet and Rocha, your family choices are Agnesidae or Ascididae. In these families that are phlebobranchs, that are solitary and have gut to the left of the branchial sac, either Agnesidae or Ascididae. Now, to tell the difference between these two, you want to zoom in onto their stigmata. Stigmata are the holes in the mesh being the branchial sac. So there are holes so that the water can flow through that branchial sac. And it helps, that water flow is going to help with obtaining food and respiration. In the Agnesidae, these stigmata, which are the holes in the branchial sac, are going to be in spiral-like configurations. As opposed to in the Ascididae, the stigmata are going to be in a straight line. So the photograph on the left, that's actually a diagram on the left, is Agnesia. So this is family Agnesidae, showing those spiral-like stigmata. And on the right, this is actually a picture of Cionidae, right, in a different family. But the reason I show this picture is because I wanted to show you the stigmata in the straight line. So in the mesh here, you see that the stigmata, which are those small rectangular holes, are in nice horizontal lines. And that's what you're going to see in the Ascididae. In the family Ascididae, there are 3 genera, and you need to look at the anterior section of the animal to be able to tell the difference between these 3 genera. And in particular, we're looking at the center portion of the anterior section of the animal. So you dissect the animal by cutting down the endostyle. So you're cutting down the ventral edge of the animal and then opening it up so you have both sides of the animal laid out in front of you. And when it's lying down like that, you've laid it down on its dorsal side to cut the ventral side. So what you're seeing is the dorsal side of the animal. And right underneath the oral siphon, you will often see a structure called a dorsal tubercle. On the left photograph here, it's this structure that kind of looks like a flower before it's opened, right? A bud of an angiosperm. So it's kind of this U-shaped structure that's nestled in this V shape here. What we're looking at is the dorsal tubercle, which is a common opening of the neural duct, right? So the neural duct is going to be the location where hormones are sent out to the rest of the body. And the dorsal tubercle is in the center of this photograph here. And then on either side of the dorsal tubercle, you see lines moving to the left and to the right. Those lines are part of the neural duct. They can also have separate openings. The dorsal tubercle is by itself an opening of the neural duct, but those bands that move left and right of the dorsal tubercle can also have their own separate openings. If there's only a single opening of the neural duct, which is going to be at the dorsal tubercle, then the genera are going to be Ascidia or Ascidiella, just like in that photograph on the left. But in the genus Phylum, there are many openings of the neural duct, which is represented in this stained image here over on the right. And in Phylum, you may not have a dorsal tubercle at all. So this, again, this neurobiological set of structures is how to tell the difference in the Ascididae, the family Ascididae, between 3 common genera. How to separate Ascidia and Ascidiella, because they both have a single opening of the neural duct. You look again at the branchial sac, and you're looking specifically where the longitudinal vessels, which are these structures that go from the anterior to the posterior of the animal, so the vertical lines, where they intersect with the transverse vessels, which are the horizontal lines that move from dorsal to ventral on the animal. In Ascidia, you have branchial papillae. You will see branchial papillae at the intersections between the longitudinal and transverse vessels. And papillae, so you see there's many box-like structures in this photograph of Ascidia, and each box-like structure embraces 5 or 6 stigmata, which are those smaller rectangular holes. Look inside each box with the darker outlines, longitudinal and transverse vessel, and you will see those little protrusions coming from one side of the box into the center of the box. Those are the branchial papillae, and those are always visible in Ascidia, but not in Ascidiella, which is the photograph on the right. So same structure we're looking at here, the branchial sac, the intersection between longitudinal and transverse vessels. In Ascidiella, we don't see the branchial papillae. In the last of the families of the Phlebobranchia that can have solitary species is the Corellidae. And they are unique in that the gut is to the right of the branchial sac. So in this photograph here, we're looking at the right side of the animal, again, because you see the atrial siphon, that's the dorsal side of the animal. The endostyle is the ventral side, so we're looking at the right side of the animal. And we're looking straight at the gut, and then the branchial sac is behind us in the photograph, in the plane of the photograph. So this is a family Corellidae. There are 3 common genera in the family Corellidae: Cheliosoma, Rhodosoma, and Corella. Fairly easy to tell apart because of the distinctiveness of the Cheliosoma and the Rhodostoma. On the left photograph, this is an example of Cheliosoma. This is the genus Cheliosoma, and it has horny plates on the tunic, as you can see represented in this photograph. There are no other Ascidian genera that have this particular characteristic. The second genus is Rhodosoma, and again, it has a characteristic that is unique among the Ascidians in that the oral and atrial siphon are protected by a lid that can go over the top, the anterior end of the animal. So what you see in this photograph is the oral and atrial siphons open and exposed. But just behind that, just ventral to that, you can see that there's another piece of body wall. And when those siphons retract, that lid closes over them. So it acts sort of like a hat, a protective covering. And that's, again, unique in the Ascidia to the Rhodosoma. And Corella is the 3rd common genus, and it has neither of these distinctive characteristics.